right? So we are now being recorded. And as I was mentioning a while ago, review is very important in order for us to get what we are studying. So that's why this video teleconference is devoted as a review also, just like our past video teleconferences on what we have covered so far. So I will not go back anymore to the midterm lesson, okay? So for example, about photosynthesis, we haven't discussed that in a video teleconference, but I will not go back anymore to that. Instead, we will start our review from the first lesson in the after midterm season. Okay. Let me just present the slide. All right, so welcome BIOL 105 YB in our 7th video teleconference. So this is our video teleconference after, I think, almost a month of not having one. So I hope you will get the hang of this again. Right, so in terms of schedule, we are actually quite on time with video teleconference number 7 because we were quite advanced in the past. So now we are back to being on track with the number of video teleconferences. Video teleconference number 7. So currently this week, we are supposed to be doing asynchronous lessons with chapter 8. So I hope you've also started viewing the videos that I posted for chapter 8 and that you've started reading as well. Okay, but our main concern in this video teleconference is a review on chapter 7, the interactions between cells and their environment. If you can remember, there was already a group discussion that I posted that I hope you've done with your group. And there's also a quiz that is open right now and it will be closed this Saturday. So I hope you're also preparing for that quiz if you haven't taken it yet. So, because you joined this video teleconference, I really hope that this will help you in your performance in the quiz because we are going to review chapter 7 right now. Okay, so as usual, I need you to be able to interact. Okay, so please use your audio if you want to say something or if you're going to answer a particular question. And if you're going to share beyond what the question is asking, that's also very welcome. So let's start the review on the interactions between cells and their environment. Before I go to the first slide, the context of the title, interactions between cells and their environment. So cells are living things, so therefore they are affected with what they are surrounded with, their environment. So it can either be the non-living environment or the environment that includes the other cells. Okay, so we have covered this in the chapter and with the videos that I've posted as well. So let's start first on what is on the cell membrane. Okay, can anyone from the group tell us what we see here?
Answer, kanang plasma membrane, tas mga proteins.
Yes, so we have the plasma membrane, the bilayer. Okay, if I can have a pointer. Right. So we have here the bilayer, the plasma membrane. Then yes, we have the proteins here, the red ones. Okay, these are integral proteins. So that's what's on the cell, okay? But what are these extensions, the green ones? Anyone?
So answer, carbohydrates.
Yes, those are carbohydrates. And collectively, the carbohydrates that are surrounding the cell is called the glycocalyx. No, so in the slides that I had in the videos, I showed you that this glycocalyx can actually manifest itself as a sticky layer no, surrounding the cell. So it helps the unicellular bacteria, for example, stick to surfaces and then move along surfaces. No, so the sliminess of bacterial colony. For example, if you haven't brushed your teeth for a long time and you try to rub one tooth with your finger, you'll notice that it's quite slimy already. No, and then there's a layer, no, kanang kit in Bisaya. Those are actually accumulated food plus bacteria colony that have stuck already on your tooth, and the feeling is quite um, slimy because those bacteria have actually also surrounded themselves with the carbohydrates. No, that's similar to this glycocalyx layer. So that's on the cell. Now we try to go beyond the cell. Okay, what's after the glycocalyx, sir? Okay, so the next topic that we have is applicable only to multicellular organisms, particularly animals, and then later on plants. No, so this is now what we call as the extracellular matrix. So in unicellular forms of life, you cannot find an extracellular matrix, okay, because they are quite independent cells. But here, we have an illustration of tissues in multicellular organisms. So first, we have the epithelial tissue and the connective tissue. Okay. Can anyone from the group tell us the difference in the extracellular matrix between the two types of tissues? How will the ECM differ between epithelial and connective tissues in animals? Anyone? Where is the ECM here? That's the yellow thing, no, that you can see here all throughout. So how do they differ between epithelial and connective tissue? You can describe it or you can give examples. Anyone from the group?
Kuan sir sa epithelial kay for anchorage ang extracellular matrix and sa connective kay mo siyang barrier between cells.
Okay, so that is correct now in your description. In the epithelial tissue, the ECM is for cell anchorage, to anchor cells. But for connective tissue, you said barrier, it's like a padding between cells, no? Maybe also a barrier, but like a padding between cells. Sort of like how cement is placed in between bricks. Diba? So the bricks will be the cells. So the name for the ECM in the epithelial tissue is the basal lamina. Okay, so the basal lamina. So as we see here, you mentioned a while ago, it's for anchorage. You can see this thin layer, and you can see the cells in the epithelial tissue. They have this anchor protein that anchors them on the basal lamina. So the basal lamina is an ECM, right? While in the connective tissue, the ECM is here, filling the gaps between the cells. No, examples of connective tissue would be tendons, your bone. Okay, that's a connective tissue. Even blood is a connective tissue. So in the case of blood, the extracellular matrix in blood is liquid. So there's a liquid type of extracellular matrix, and that's the plasma in the blood. Okay, which fills the gap between the blood cells. All right. So What else can be seen here? What are these red fibers here, and then what are these black stuff in the epithelial tissue? Anyone?
Sir, ko answer. Ang red kay muna ang cytoskeletal filaments.
All right. Yes, they're part of the cytoskeleton. Correct.
Ang black sa side kay muna ang cell junction. Cell to cell junction.
What type?
Ay, wala ako
Correct.
comment, sir.
Okay, you're correct. Go, continue. What about the one at the bottom?
Sa bottom, sir, muna ang hemidesmosomes. Ang para cell.
Yes. Yes. So all of the things that you said were correct. Okay, so later on we will go deeper into that in the later parts of the slide. So thank you. All right, so next. So speaking of the ECM, there are four main components, no, or molecules found in this ECM, and one that's attached to the cell. Okay, so first we have collagen, and then we have proteoglycan, and then fibronectin, laminin, and integrin. So in this illustration, you can see a simplified overview of all of these components, no? So look at the shape of the collagen. Sorry, as shown here. Diba? So ang collagen, ito yung fibrous na made up of alpha helices, as you can see here. And look at the shape of the laminin, di ba? Yung cross-shaped molecule, that's the laminin, okay? And then this one that looks like two segments attached together on one side, that's the fibronectin, di ba? Okay. And then we have this parang frond-shaped molecule with parang may mga fronds or leaflets. These are the proteoglycans. And of course, the one attached to the cell here that exists as a pair, no, is the integrin. Okay, very important in communicating what's on the outside to the inside and vice versa. So let's take a look at each of these. Can anyone from the group tell what is shown on the slide?
Collagen, sir.
Collagen. So can you elaborate on what's shown on the left and what's shown on the right? Anyone, or you can say it yourself.
One, sir. Sa left kay mo ng pagka-arrange sa collagen fibers in the cornea sa itong mga mata. And kanada yung sa right kay type 4 collagen na network.
Yes. All right. Where can we find this type 4 collagen? Anyone again? This is found in the epithelial tissue. Which part of the epithelial tissue?
Sa basement membrane, no, sir?
All right. Basement membrane or the basal lamina. No? That's the ECM of the epithelial tissue. That's where you find type 4 collagen. Then this one sa mata, sa cornea, I believe these are type 1 collagen, but they are arranged so that they actually appear transparent, okay, so that light can pass through the cornea. So can anyone from the group again tell us the structure of collagen or its properties? Anyone? Structure or properties?
High tensile strength, sir. And I think may siya helix, may composed siya of alpha helices.
That is correct. So a collagen molecule is resistant to stretching forces, no? So on your skin, di ba, your skin is epithelial tissue, may epithelial tissue dyan. The collagen there help in keeping your skin flexible, no? So when you stretch it, it goes back to its original shape. So that's when you are still young, no? But when you get older, the collagen fibers are also sort of eroded. They become weaker. So that's why older people, their skin are less elastic. Okay, so that's collagen. And it's made up of the helices, no, the alpha helices that are intertwined together. And then we have this. Can anyone from the group tell us what this molecule is and what its properties are?
Kuan na, sir. Proteoglycan.
Yes, that's correct. Can you say something about its structure or properties?
Kuan, sir. Kana iyahang mura siyag centipede, sir, kay na siya main arm, and then on the main arm na uban arms pa na made of core proteins, and then ang core protein na arms kay naa sila yung mga subpagid na arm na kanang nasa screen, sir, kanang chondroitin sulfate o katong keratan sulfate, and then tungod sa ilahang sulfate groups ba to, sir, o
Yes.
basta ionic groups, kay mag-attract sa water molecules busa na siya high compressional strength.
All right. So everything you said was correct. No? So the main structure of the proteoglycan is the one shown on the right. So ito. So you have the bigger central molecule, the hyaluronic acid. And from that, there are core proteins that are emerging from it. So the center here, the black line is a protein. And then the red lines emerging from it are carbohydrates. So that's why the name is proteo, meaning protein, and then glycan or carbohydrate. That's why they're called proteoglycans. And the carbohydrates are all containing the sulfur ions that we see here. So that's the reason why they attract water, and the water fills the spaces between the arms that we see here. And as you mentioned, because of that, they have high compressional strength, resistance. So in your ECM right now, say in your skin, it's not just collagen that's present. Diba? So collagen has a high tensile strength resistance. There are also proteoglycans which complement the strength of the collagen. So the presence of proteoglycans in your connective tissues allow your connective tissue to withstand compressing forces. Right? So that's the proteoglycan. And then next we have this molecule. Anyone from the group tell us what this is and describe it?
Kuan na, sir. Fibronectin.
Okay, correct. How do you describe the structure or what is the function of this? The structure muna.
Ang structure niya, sir, kay sa murag two chains na attached in one end by disulfide bond, sir.
All right. So that's correct. Um, In which particular stage of life are fibronectins very important?
Kuan sir, usually sa embryonic stage.
Yes, why?
Kay murag yung main function is for cell migration and for proper formation of glands.
All right. So the presence of fibronectin will allow the cell to move um, onto that surface. No, so during developmental stages, many cells actually migrate from one point of the embryo to another, and they can do that. They can track where they're supposed to go through the presence of fibronectin present on surfaces of the embryo. Okay, so as we will see later on, fibronectins actually also have this RGD sequence, as we will see now because it's already here now. Okay, can anyone tell us what the function of this RGD sequence on the fibronectin is? RGD stands for arginine, glycine, and as I think this is aspartic acid or aspartate. So, what's the function of this RGD sequence?
Kuan siya sir for integrin binding.
Yes, so as you can see here, this is where it's found, the RGD sequence. It's for integrin binding. So that's very important, no? Because if we go back to the other slide, if I may just go back here, look at the fibronectin here. Okay, itong violet. It's bound to the integrin, and it's bound to the integrin because it has an RGD sequence. Okay, and that's one way of the cell clinging onto its extracellular matrix through the binding between the integrin and the fibronectin. Okay, so that's the fibronectin. So here, what do we have? What do we have here? Anyone from the group? Can we find this in the connective tissue or epithelial tissue?
Sa unsa na sir sa epithelial?
Yes, this is from the epithelial tissue. So again, can you describe to the group now what are these green molecules and then what are these pink long molecules?
Muna tong unsa sir mga laminin na molecules ang na murag nag intertwine siya sa collagen na fibers kanang pink.
Yes, so that's correct. No, so you have the laminin, the cross-shaped molecules that are attached to the a type 4 collagen present here in the basal lamina. So if I may go back to the previous slide, where is this located? It's actually located here, this basal lamina that you see here, that thin layer that's made of the type 4 collagen and the laminins that are attached together. Okay, so that's this figure in context. All right. Um, then we have this illustration. What is happening here and what are the chemicals behind this process? So you can see here the basement membrane or the basal lamina, the red line. You can see the connective tissue here at the bottom, and then the orange layer here on top would be the epithelial tissue. And then we have these pink cells. They are connective tissue. What's happening here specifically? What cells are these? The pink ones. Are these normal cells? Okay, anyone? I think you already know what it is, okay, just by looking at it.
Kuan na, sir? Cancer cells?
Yes, okay, these are cancer cells. And can you describe what's happening in
Kuan
the
na, sir,
pictures or drawing?
metastasize.
Yes, okay, so they are currently in the process of spreading to the other parts of the body called metastasis. Um, how do they do that? What is in them that allows them to do that? One aspect in terms of ECM, extracellular matrix.
Kuan na to, sir, na sila excess production of atong matrix metalloproteinases.
Yes. Okay. So cancer cells take advantage of the enzymes known as matrix metalloproteinases or MMPs. What do these enzymes do? They break down extracellular matrix. No, so that's the function of the enzyme. And the cancer cells have excess na nubra ang ilang MMPs. So they use that to their advantage to digest or break down the basement membrane that you see here, and they can enter through the basement membrane, no, into the connective tissue. And in the connective tissue, they can further use their MMPs to break down the connective tissue so that they can pass through. Okay, but MMPs normally function in tissue remodeling. No, just like this is how I explained it in the video. Just like when you build a house, you want to renovate, you want to extend the wall, you need to break down the wall first before you can extend it. So in tissue remodeling, say in the healing of wounds or development of tissues, no, or organisms, the MMPs are the equivalent of the hammer and the chisel that will break down the wall. So cancer cells have an excess of these. So ang cancer cells, they can destroy extracellular matrix even out of the context of development. And they use it to their advantage to spread to the other parts of the body. So please remember that one. I believe that will come out in the quiz, no? MMPs. Question. What are the important cofactors in order for MMPs to work? These are ions. What ions are these? The cofactors.
Kuan, sir. Sa to, sir, zinc and calcium.
Yes, all right. So zinc ions and calcium ions, to name a few. I think those are the most important cofactors for MMPs to work. So MMPs need a cofactor because they're enzymes. All right. So remember that one. That will come out in the quiz. I'm telling you because you're attending this. So yung hindi naka-attend, sorry na lang unless they view this video. All right. Now, we have discussed the four. Okay. Now, the four components of the um, ECM. Now we have this, the one attached to the cell. How do we call this molecule, the one that we see here? The green one.
Ano siyang integrin, sir?
All right, so these are the integrins. They are dimers. They're composed of alpha and a beta subunit. When they are inactive, they are sort of bent down and they are sticking together. Okay, and the blue layer here is the plasma membrane. So they are integral proteins. But once they activate, they actually lengthen, and each of the ends of the integrin dimer can bind with certain molecules. So in this case, on the outside, it's bound to collagen. On the inside, it's bound to tallinn. Okay. Um, so that's when they are activated. So there are two ways on which they can be activated. Either the signal is from the outside or the signal is from the inside of the cell. So they're also known as integrins because of that reason. No? They can integrate what's happening on the outside with what's happening on the inside of the cell. And they're integral proteins. So um, now that we have had a rundown of all the components of the ECM and the integrins, let's look at some of their functions, no, together. First, we have this illustration. Can anyone from the group tell us what's happening in this illustration? So we have here, I think this is number 1, number 2, number 3, and then number 4. So it's arranged that way.
Kuan, sir, ang cell kay...
This is the cell, no? This is a cell. So what is this? Continue.
Ay, kuan siya, sir. Ang cell kay hinihinay siya ga-anchor sa surface na non-native siya o foreign surface.
All right. So this cell is placed on a non-native surface or a glass, perhaps in a Petri plate, and it's slowly anchoring itself, no, onto the surface. Okay. So how can it do that? What particular molecule in the extracellular matrix, no, allows the cell to do this? Is it the collagen? Is it the fibronectin? Is it the laminin? Is it the proteoglycan? Is it the integrin? Which one?
Integrin, sir.
All right. So the integrin is like, I described this in the video. It's like the tube feet of a starfish. Diba? If you put a starfish on a rock, the tube feet will attach on the rock. So the integrin is like that in sort of in function. No? So we can see here an illustration of the cell with the integrins color coded as orange, and attached to the integrins are the actin filaments. So the actin filaments are like the arms, and the integrins are like the pincers that hold onto the surface. Okay? And how do we call this type of adhesion? There's a name for this.
Focal adhesion, sir.
All right, so this is focal adhesion, okay? And the cell does this so that it will be able to hold on to a particular surface that is not its native environment. No? So think of yourself riding a roller coaster. In order for you to not fall off the roller coaster, you need to hold on to the railing with your hands. So that's how the cell is doing also here. Your hands are the integrins holding onto, and then your arms that are attached to your hands are the actin proteins that help with the force, no, of you holding onto. So you can analyze it that way. So here's an illustration of that. You can see here thousands of integrins thousands of integrins that are holding onto this glass surface. And then attached to the integrins are these. No? So the blue ones are the actin filaments, part of the cytoskeleton. And then we have these. Let's talk first about the hemidesmosome first, the one here at the bottom, the one that's also pictured here. Can anyone from the group tell us the function of the hemidesmosome?
Conscious sir for cell anchorage sa native niya na location.
All right. So what is that surface that it needs to anchor itself on? How do we call that? Unsa man tong nga surface? This one. What is this surface? This blue layer.
Basal lamina, sir.
All right, the basal lamina. So the hemidesmosome is usually found in cells that are, nahaman makita ng basal lamina, that are part of the epithelial tissue. Okay, that's where the hemidesmosome is. And notice here, you can see the type 4 collagen. Dili day. That's a type 4. The type 4 are the blue ones here. Okay. And then you can see these cross shape. That's the, what are these cross shape things? The laminin. Okay. And then you can see here the green shapes. These are the integrin. No, and they are holding together onto each other so that the cell can be attached onto the basement membrane. Okay. And then on the other side, you have here the other proteins like plaquins, no, or the plaque proteins. And then at filaments that you see here. Okay, so in context, this is where it's located, not the bottom of the cell here. That's where the hemidesmosome is. All right. Now let's talk about the other junctions. Okay. Anyone from the group choose a junction and say something about it. So we have adherens, we have gap, we have tight, and then we have desmosome. Anyone from the group?
Kuan sir. Ang isa sa junctions kay tight junction. Ang tight junction sir kay
All right.
junction siya which does not allow any fluid to pass through.
Yes, it doesn't allow any fluid to pass through in between the cells, no? So in between the cells. And that's why it's called tight junction. You're correct there. What proteins help in the formation of the tight junction? How do we call those proteins?
Kuan sir. Claudine.
Yes, so Claudine. So the way I remember this is they block, no? So like how clouds block the light from the sun. So you can remember it that way. So they're known as Claudine. Okay. Next, another junction, please. Anyone? Anyone?
Kuan sir napay isa na unsa sir. Uh, Plasmodesmata.
It's not shown here yet. We are
Oh,
going ahead
yeah.
of ourselves. Okay, ito muna.
Ah, kuan sir. Gap junction.
All right. Okay,
Ang gap
go.
junction kay composed of proteins called connexins which aggregate to form connexon. Pag mag-align tayo na sila, sir, mag-form na siya og passageway from one cell to another.
Okay, and it serves as a communication channel between adjacent cells, no? So that's why it's also known as a gap junction because it provides a gap as a passageway for communication between cells. So that's correct. So we have two left, desmosome and adherence junction. If you look at them, they seem to have the same purpose, no? It's for adherence or adhesion between two cells, two adjacent cells. Can anyone in the group tell us how do they differ? Adherence and desmosome. Or you may just want to describe one of them. That's fine.
One sir, sa adherence junction, ang ga-adhere sa ila between cells is the cadherin, which ang naka-attach sa cadherins sa sulod sa cell kay ang catenins, which ang catenins po kay ga-attach po siya sa actin filaments, while sa desmosomes kay ang ga-adhere sa between cells kay ang desmoglein o desmocolin, which na instead na actin filaments ang ni-attach sa sulod sa cell kay ang intermediate filament.
Everything about that is correct. They have sort of the same function. But notice also here the positioning. This is important, no? Ang adherence junction is positioned here on this upper portion of the cells, no, on this layer. But then the desmosome are positioned here near the bottom where the basal lamina are, no? And the reason why these are actin filaments because actin filaments are quite stretchy and they can move, no? So the cell can easily adjust its position when the tissue is sort of moved. So that's the function of the adherence junction and the actin filaments. While the intermediate filaments are somehow stiffer, so mas stiff sila, and they somehow resist movement more than actin filaments. So they provide resistance to movement here near the bottom of the cell. All right. So you may also notice that desmosomes have very similar structure as well to hemidesmosomes, but desmosomes attach cell to another cell while hemidesmosomes attach a cell to a basement membrane. Okay, so good. Next. Okay, can anyone from the group describe what's happening here and what's behind this phenomenon? Or I will describe what's happening. Okay, so here, this is embryonic development. No? So we have a layer of cells that have not differentiated. But then here, we can see that some cells have differentiated. They have different color, obviously. And then we notice that they have sort of separated themselves from the original group. And here in the final one, they have formed their own group to form the neural tube. So what is the mechanism? What is behind the cell recognizing that they are not anymore in the same group?
Ang cadherins ba, sir?
Yes. So it's the cadherence. And what's the function of cadherence?
Kanang cell to cell adhesion, sir, between cells.
Which types of cells can adhere to each other?
Same type, sir.
They have to be what? Same type, no? So if this may help you, you can look at the word cadherin, no? Adhere, stick. What's the C? It could mean co, co-adherin, di ba? The same type of cell. So parang co-lig, cadherin, adhere between the same type of cell. So what's happening here is that the blue cells here are expressing different cadherence now from the orange cells. And so they recognize that and they separate themselves because they cannot adhere anymore with the same cadherence. And instead, the blue cells are adhering to their co-blue cells because they express the same cadherin. And the orange cells here adhere with their orange cells because they also express the same cadherin. That's very important, and I think it will come out in the quiz. All right. So we're now done with this. Okay. So kanina, someone mentioned this. So can anyone describe the illustration shown on the slide?
Ay, ko answer mo na natong plasmodium mass.
Yes. So what is the function of a plasmodesma? Desmata.
Sir, sir, channel between the cell wall of two adjacent plant cells.
All right. So they are sort of the equivalent of your gap junction, di ba, in animal cells. But this time between plant cells, no, which are surrounded themselves by cell walls. So here in the illustration, you can see here the line here. This is actually the plasmodesma, singular plasmodesma. And here we have another plasmodesma. Okay, so plural plasmodesmata. So communication between two adjacent plant cells. And last but not the least, a special type of extracellular matrix is the cell wall. Okay, so the cell wall, as we know, is composed of cellulose, but here we are looking at it, at its components, we can see that there are hemicellulose, the main rods here, and then perpendicular to that, these are, what are these? This could be the pectins. So we have pectins pala here. And then here the glycoproteins. Okay. And then, sorry, hemicellulose are these. Ito yung purple ones. And then the cellulose microfibril are the big ones. Okay, sorry, I switched them up. So that's the structure of the cell wall. No? So it's like a concrete composed of crisscross components of cellulose microfibril, the main one, and then the hemicellulose, and then the pectin and the glycoproteins. So how is this made? It's made through an enzyme called cellulose synthase. No? And the cellulose synthase only makes the cellulose microfibril, the big ones. What about the smaller ones here, the other components? How are they made? Can anyone answer? Again, the cellulose synthase makes the cellulose microfibril, the big rods here. How about the smaller components like the hemicellulose, the pectin, and the glycoprotein? How are they made?
Sa Golgi body basta siya, like synthesize siya ay
Ito,
paghuman o
continue.
uh ano siya after the cellulose synthase kanang synthesizes the microfibrils, the cellulose microfibrils. The other components na then follows, which came from the Golgi complex from inside the cell para to synthesize the complexity na then of the cell wall.
That is correct. No, in every sense. So the rest, aside from the cellulose microfibril, are made from inside the cell, no, from the ER and then to the Golgi complex. and then deposited onto the already formed microfibril which the cellulose synthases make. So, bale, kung sa construction pa ng building guys, think of the microfibril as the kabilya, di ba? You're familiar with the kabilya. So, kabilya muna. And then you add the hollow blocks and then the cement later on, and then you have your final wall. Okay, so that's how the cell wall is made. Right. So just some reminders before we end the video teleconference. So the quiz on chapter 7, the one we just reviewed, is still open. It will close on March 13, Saturday at 8 p.m. Okay, so at your convenient time, please do take the quiz. The group discussion for chapter 7 is posted. I believe nag-due naman siguro this one. So I hope you were able to discuss with your group or fill out the tables, no, for that. So with that, are there any questions or concerns? Questions, concerns? Your midterm grade is not yet finished, so you might have to wait a little bit more before you can see it posted in the SLMIS. Questions, concerns?
And so far, sir.
Right. Next week, no, in this video teleconference, we will discuss chapter 8. So I encourage you to also be present and tell your classmates to be present because we are going to review. Later this afternoon, I will meet you because we will have a video teleconference for our lab, no, YB. I will be discussing there, of course, a review of DNA extraction and then PCR as well. So I'll see you this afternoon, guys, and I thank you very much for joining this video teleconference. So I hope you'll have a productive day ahead, and you are now all dismissed.
Bye,
Thank
sir.
you, sir. Stay
Thank you,
safe.
sir.